Alright, so previously I ended the first segment with kind of going over how exactly it works. Now I'm jumping right back into it, working with this reporter plasmid. So how exactly did I go about making it? So the first step was to insert these cleavage domains at various sites to see if the insertion of these cleavage domains would have an impact on luminescence. Because if you insert these things and it changes the protein, the nature of the protein, the structure of the protein such that it doesn't work, then the whole thing would just stop working. It wouldn't be worth doing. It wouldn't function as a reporter plasmid. So we had to test this first. Here you can see a graph with the different sites I checked. So poly B, these are controls. BSRG1 is one of the sites we used. SPH1, XCM1, and AVA1 these are 4 different sites. BSRG1, we started using it right off the bat, and it gave us some more consistent results, which tended to have higher values than the rest of these. So BSRG1, or the location BSRG1, was the site we decided to go with. So the next step was to insert my cleavage domain at the BSRG1, causing a frame shift mutation which truncates the protein. So like I kind of said earlier, depending on how you insert this cleavage domain, you can truncate the protein, which effectively makes it like a third or the fourth of its normal size, which means that it won't work. So that's what I went about doing. So after I did that, I had my BSRG1 reporter plasmid in frame, which with the protein not truncated, and my BSRG1 reporter plasmid with a frame shift truncating the protein. So here was just kind of an experiment to compare these two proteins, or these two reporter plasmids. As you can see over here on the far left, this is the BSRG1 protein in the absence of the endonuclease. Here is the BSRG1 in the presence of the endonuclease, which is, you see a drop in the luminescence, which is what we expected to see. Here is the BSRG1 frame shift reporter plasmid. And as you can see, the numbers are close to 0, which is what we wanted to see, actually. We wanted to show that the frame shift would cause an extreme reduction in luminescence. And then here is the BSRG1 frame shift in the presence of the endonuclease. So kind of how I went over earlier, if it's in the presence of the endonuclease, it can come in, and if the non-homologous end-joining pathway is working, then you should see an increase in luminescence, which is exactly what we saw. So these results seem to indicate that the plasmid works, or the reporter plasmid works. So the next step is the assessment of this repair pathway in AAG2 cells, which, if I didn't mention earlier, are mosquito cells, embryos, and mutant embryos. So right off the bat, we kind of got this. We had this reporter plasmid. We want to see if we can utilize it for its intended purpose. So a couple of notes: double-stranded RNA is used in RNAi knockdown. Now, RNAi is an immune pathway, which is typically found in eukaryotes, which allows eukaryotic cells to defend against viruses, which can take the form of double-stranded RNA. So a couple of years ago, I think it was close to a decade ago, scientists found out that you can utilize double-stranded RNA to knock out genes of interest. So you can utilize their own immune system to remove the function of proteins temporarily. So keeping that in mind, Q70, which is a gene or a protein which is suspected to play a role in non-homologous end joining. So it's a gene which we thought plays a role in the cellular repair. And as you can see here, we have our reporter plasmid with the endonuclease with double-stranded RNA targeting GFP, which is our control. It doesn't really actually target anything. And here's our reporter plasmid, the endonuclease with the double-stranded RNA targeting Q70. And as you can see, there's a significant difference, which suggests that by adding this double-stranded RNA, which targets Q70, you see a reduction in non-homologous end joining activity. So that's exactly what we wanted to see, and it shows that the reporter plasmid is working. So here are my planned experiments, or where I'm going to go on with this specific portion of my project. Specifically, I'm going to assess the non-homologous end-joining repair pathway in cells and embryos utilizing RNA-mediated knockdown of key components, like the previous slide. And then I'm also going to assess the pathway in mutant embryos which are deficient in key components. So instead of knocking them down temporarily, we knock them down permanently, so they're gone. And then we can test them that way. And some targets for knockdown and knockout are 270, which I mentioned earlier, and ligase 4, which is just another protein involved in non-homologous end-joining pathway. I'm also doing a lot of reading, trying to find some alternative targets for to test effectively. And analysis is done using said statistical analysis. So my second objective involves the genetic manipulation of ASGPA. So currently, if we want to genetically manipulate ASGPA or create a transgenic or a knockout line, we do embryonic injections. So embryonic injections take a lot of time, they're laborious, they're just all around. It's time consuming, requires a lot of training. People that do them hate doing it, and they spend days and upon days injecting thousands of embryos just to get a handful of positive results. So what I'm working on is a new technique which, instead of using embryonic injections, utilizes adult female injections, which will hopefully save money, time, requires less training, and shows or allows to benefit from a generation skip. So what exactly is a generation skip, and why is that important? So up here you can see our typical embryonic injections involve the injection of an embryo, which is considered your G0 embryo, your generation 0 embryo. So you inject it with your effector plasmid or your protein, whatever is going to cause the change that you want to see, and in about 2 weeks you get your generation 0 adult. So the generation 0 adult you then cross or mate with your parental strain or whatever you want to mate it with, and then in about 1 week you get your G1 embryo, which is the end goal. It's your generation 1 embryo, it's your mutant, your knockdown. That's what you did the whole process for, is the G1 embryo. Now with adult injections, you inject the actual female adult with your effector plasmid or protein, and in about 1 week you get your G1 embryo. So you cut out 2 weeks worth of work and time, which is, depending upon how many genes you want to look at, so that's 2 weeks per gene. That's a lot of weeks and equals a lot of money. So this adult injections technique would be extremely beneficial. Which brings me to objective 2, which I'll probably cut short just so I can include all of this in the last segment of the video.